Here we come to a big concept with a very important sounding name because it's very important. This is what we call the central dogma of molecular biology. The central dogma says that information is transferred from DNA to RNA to protein. We see it on your guided notes. You can see a diagram that shows the same thing. What does the central dogma imply about the role of RNA? Well, RNA is very important. Even though the genetic information is all coded on DNA, we know DNA is important because it passes traits from parents to offspring. We know proteins are important because proteins do all kinds of stuff in cells and in living organisms. But the RNA is also super important because without RNA, you would not be able to transfer that information from the DNA to the proteins. So the DNA contains a lot of code for making proteins, but it's the RNA that does the work, that reads that code on the DNA and then transcribes it and translates it into proteins. The central dogma has some limitations. It does not represent the other roles of RNA. There are other roles of RNA. RNA does a lot of stuff. There are also exceptions to this dogma. For example, there are viruses that contain RNA and then they infect organisms by transferring their information from their RNA to the DNA of the host organisms. Consider this illustration of gene expression in this slide. DNA, RNA, and proteins are all involved in putting genetic information into action in living cells. This illustration is a simplification, and there are other things that we've learned about how genes are expressed. But when scientists talk about a gene being expressed, like we've learned about dominant and recessive alleles and what kind of gene will be expressed in the organism, but really when scientists talk about a gene being expressed, what that means is that that gene is being used to make a protein through transcription and translation. So an expression of a gene is indicated by the formation of a protein coded by that gene. Expressing a gene means that its code was used to build that protein. DNA carries information for specifying the traits of an organism. The cell uses the sequence of bases in DNA as a template for making mRNA. The codons of mRNA specify the sequence of amino acids in a protein. Proteins, in turn, play a key role in producing an organism's traits. This helps explain the connection between genetics and molecular biology. Many RNA molecules are not translated into proteins, but they can still play important roles in gene expression. Here we see the DNA strand. Remember that is replicated, so it can copy itself. It can be copied within the nucleus. But in the central dogma, the DNA strand is used as a template to make information, to make messenger RNA. The information from the DNA is copied onto the mRNA on a complementary strand of mRNA. After the mRNA is formed in the nucleus, it travels through pores in the nucleus into the cytoplasm, and then in the cytoplasm, it finds ribosomes. There are a lot of ribosomes floating around in the cytoplasm. Some of them are floating around, some of them are actually attached to other structures. But the mRNA will find one of these ribosomes, and then the ribosome will attach to the mRNA and start reading it and translating it. The ribosome, which consists partly of ribosomal RNA or rRNA, will translate the code in the mRNA into a sequence of amino acids that will be joined together to form a polypeptide chain. The polypeptide chain, remember the ribosome starts reading the mRNA at a start codon, AUG, and it will stop reading the mRNA at a stop codon. So when it gets to a stop codon, it will stop making that polypeptide chain. It will release the polypeptide chain, and the polypeptide chain will finish forming a protein as it will fold over itself. One of the most interesting discoveries of molecular biology is the almost universal nature of the genetic code. Although some organisms show slight variations in the amino acids assigned to particular codons, the code is always read 3 bases at a time, and it's always read in the same direction by ribosomes. Despite their enormous diversity in form and function, living organisms display remarkable unity at life's most basic level, the molecular biology of the gene. And this great unity helps scientists develop evolutionary relationships between organisms. So plants and animals that all use the same genetic code or similar genetic codes are more closely related than prokaryotes that use different genetic code. And this tells us that the prokaryotes and eukaryotes diverged very early on in their history. And by studying genetics, we can see at what point in the history of each lineage, different evolutionary lineages branched off to form, to continue to evolve separately from each other.